Welcome to This Explainer. Okay, today we are diving into how our bodies build, well, literally everything from the ground up. Right at this very second, like right now, inside almost every single one of your trillions of cells, there's this microscopic cellular assembly line just humming away. It's a completely elegant, highly organized manufacturing process that actually takes a chemical code and turns it into physical reality. By the end of this, I promise you're going to see your own cells in a completely different light. But before we talk about the human body, let's take a quick trip to Asinara. It's this small island off the coast of Sardinia, and this island is famous for something pretty wild, a population of completely albino donkeys. Just look at that stark white fur, the pink eyes, the hooves. So why exactly do they look like this? Well, it all boils down to one tiny microscopic error on that cellular assembly line we just talked about. One tiny error that completely alters their whole physical makeup. We're going to solve this biological mystery today by uncovering exactly how our genetic code creates physical reality.
Here's our roadmap for today's explainer. First, the cellular assembly line. 2, transcription, which is copying the blueprint. 3, translation, building the product. 4, we'll compare factory layouts. And finally, 5, what happens when the assembly breaks?
Let's kick things off with part 1, the cellular assembly line. You know, every factory needs workers, machinery, and a management team, right? Well, your cells are absolutely no different. The central concept driving this whole microscopic factory is called gene expression. Simply put, this is the process by which DNA directs the synthesis of proteins. You can kind of think of proteins as the physical bridge between your genotype, which is your hidden genetic code, and your phenotype, your physical observable traits. If the DNA is the big idea, the proteins are the physical reality.
To actually pull this process off, we rely on a core crew of 3 main players. It's a great rule of 3 to keep in your back pocket. First up, we've got DNA. This is the master blueprint. It contains literally every instruction needed to build you. But here's the catch. That master blueprint is way too precious to risk getting damaged out on a chaotic factory floor, so it actually never leaves the protective vault of the nucleus. Because that DNA stays safely locked away in the biological front office, we absolutely need our second player, RNA, specifically messenger RNA. Think of this as a disposable, photocopied set of instructions that can be safely sent out to the factory floor. And third, we have the ribosomes. These are the heavy-duty manufacturing machines. They're the workstations that actually read those photocopied instructions and physically assemble the final product, which is a protein.
Moving right along to section 2, transcription, or copying the blueprints.
Okay, so this entire first phase takes place in that biological front office we mentioned, the nucleus. Now, the cell doesn't just blindly copy everything, it targets specific genes using a special region called the promoter. In eukaryotic cells like ours, this promoter features a super crucial sequence affectionately known as the TATA box, because literally it's just the sequence of nucleotides TATA. Watch how specialized proteins called transcription factors spot this exact TATA box and carefully dock onto it. Only once those factors are perfectly positioned can the big boss enzyme RNA polymerase II finally bind to the DNA. Together, they form this whole transcription initiation complex, which allows the DNA double helix to essentially unzip, and the copying begins.
Once it gets the green light, that RNA polymerase enzyme just takes off, traveling downstream along the unwound DNA. It acts exactly like a zipper, untwisting that double helix just enough to expose the DNA nucleotides so it can pair them up with complementary RNA nucleotides. And it builds this messenger RNA strand fast. We're talking a brisk pace of about 40 nucleotides every single second. As the enzyme moves forward, the newly minted RNA strand peels away, and the DNA behind it, it just neatly zips right back up. Eventually, the enzyme hits a specific termination signal, cuts the new RNA transcript completely free, and detaches. Boom! The photocopy is officially complete.
All right, into section 3 we go, translation, building the product.
So now that we've got our messenger RNA, let's follow those newly minted instructions out of the front office and right onto the bustling factory floor, which in the cell is the cytoplasm. Now, you might be wondering, how on earth do we translate the 4-letter chemical language of RNA into the language of proteins, which are built from 20 different amino acids? Well, the math actually gives us the answer, and that answer is 64. You see, the genetic code is read in triplets, little 3-letter words called codons. Because we have 4 possible RNA letters and they're arranged in groups of 3, the math works out to 64 possible combinations.
That is more than enough to specify the 20 amino acids we actually need. And honestly, this beautifully illustrates just how mathematically elegant the genetic code is. It's got built-in redundancy, meaning several different codons can actually ask for the exact same amino acid. It's like a built-in safety buffer against tiny errors.
But who's actually down there reading these three-letter codons? Enter transfer RNA, or tRNA for short. I want you to think of tRNA as a tiny factory forklift. On one end of its weird L-shaped structure, it carries a very specific piece of cargo, an amino acid. On the exact opposite end, it has an anticodon. This is a three-letter code that perfectly matches and just magnetically clicks right into the corresponding codon on the messenger RNA. So when the forklift spots its matching code, it knows exactly where to drop off its amino acid payload.
Now, all of this frantic forklift activity, it happens entirely inside the ribosome, our main manufacturing machine. And what's really cool is how this machine is organized into three specific operating stations. First up is the A site, which conveniently accepts the new tRNA that's delivering the next amino acid. Next is the P site. This one holds the tRNA that's attached to the growing polypeptide chain. This is where the pieces are literally linked together. And finally, the E site, which is where the completely empty, depleted tRNA exits the machine to go hunt down another amino acid. Accept, hold, exit. It's this continuous rapid-fire assembly line, linking amino acids together at just incredible speeds so they can fold into a final functioning protein.
Let's transition to section 4, comparing factory layouts. Depending on the organism, this factory floor plan can actually look pretty dramatically different. Take prokaryotes, like bacteria, for instance. They operate a lot like a super streamlined one-room workshop. Because they literally don't have a nucleus, there are zero walls, which means the ribosomes can just jump right onto the messenger RNA and start translating it while it is still being transcribed. It happens perfectly simultaneously. On the flip side, eukaryotes, like plants, animals, and us, we have a highly segmented factory. The nuclear envelope creates this strict physical barrier. So transcription happens exclusively inside the nucleus, and translation waits to happen out in the cytoplasm. And this segmentation is a real game changer. It allows us to extensively edit and modify our RNA instructions before they ever hit the factory floor. It's a massive hallmark of complex life.
And finally, section 5, when the assembly breaks. So what exactly happens when there's a typo in that master blueprint and the cellular machinery totally malfunctions? Well, even a single wrong letter can just cascade disastrously through the entire factory. Way back in 1902, a British physician named Archibald Garrod was studying genetic disorders, and he actually coined the phrase, inborn errors of metabolism. He was literally the very first person to suggest that an inherited faulty gene leads directly to an inability to make a specific functioning enzyme. Basically, if the factory produces a broken machine part, the entire metabolic pathway comes to a screeching, grinding halt.
And that brings us perfectly back to those wild Asinara donkeys we talked about at the beginning. The absolutely crucial point here is that these donkeys aren't magically painted white. They actually have a tiny genetic mutation in their DNA blueprint. That single typo gets transcribed into messenger RNA, and the ribosome dutifully translates it just like it's supposed to. But the resulting protein, an enzyme called tyrosinase, is completely faulty. Because this one specific enzyme is broken, the entire metabolic pathway that produces melanin, which is the dark pigment for fur and eyes, completely stops. So the mystery is solved. A microscopic typo on the cellular assembly line creates a dramatic whole-body physical change.
Think about this. Every single second of your life, in almost every single cell of your body, this elegant, complex dance of DNA, RNA, and ribosomes is taking place. Millions of tiny assembly lines are just churning out the proteins that keep you alive, breathing, and thinking. So if one microscopic typo can change the color of an entire organism, just how perfect, how staggeringly precise is your own genetic machinery humming along inside you right now? Thank you so much for joining me on this explainer. Keep learning, keep wondering, and I'll catch you next time.